My name is Arutha Kulasinghe. I'm based at the Frazer Institute. This is our second year of cure cancer funding, and today I'll be talking to you about some of that work that's being done in sunny Brisbane. And so I thought I'd talk about, our motivations for this. This is Michael Lytell, a lung cancer patient advocate, non-smoker, non-drinker, EGFR mutant, stage 4 lung cancer, who advocates for research around the world. And with Annabelle Gurwitch, she's a Hollywood actress, went for a COVID test and found out she had stage 4 lung cancer. And so she's in the Michael Bay movie, Ambulance. She's the psychologist on that. And Michael, he's a great advocate for cure cancer and a lot of the work that we do. And so luckily we're able to, go to conferences with Michael and other patient advocates like this. And we often argue about, who we look like from the bad boys. And so that's our motivation to this work. And so my lab really focuses on spatial transcriptomics, proteomics, interactomics. You would have heard these terms over the last couple of years, but it really took Eric Topol to name the field spatial medicine. This is where the field's really heading. We are understanding every cell within a patient's biopsy, and this is truly transformative technology developments if we can find the right applications. And so Nature came out earlier this year and called it tumor geography. Lots of terms around this, but it's really getting that visual information simultaneously with your molecular readout. So immunotherapies, we've heard a lot about this. Our focus is in lung cancer, where a subset of patients respond to these therapies. Our goal is to really understand what is the underlying tumor microenvironment driving these responses. And so this article came up a couple of years ago and showed that tissues and not blood are where immune cells function. I did my PhD in the liquid biopsy space, and so I had to transition quite quickly to really understand fundamentally what's happening at a tissue level to then inform on what to look at in the blood. So there are a couple of predictive biomarkers. We've heard about this, but what my lab really tries to do is to fundamentally profile every cell within a patient's biopsy. So 3 to 5 million cells per surgical resection or biopsy sample. And why this is important is if we were to grind these tissues up, from the early 2000s, we'd get this average level expression for these markers. Single cell transcriptomics, proteomics approaches really like going into a car showroom and seeing all the parts on the floor, but not being able to see the car for the car. And so spatial transcriptomics and proteomic approaches are really allowing us to see the tissues for where they are in the state in which they're found. And so if you look at these two tumors on the bottom left, the tumor on the right is highly infiltrated compared to the one on the left. But if we were to look at it at a bulk expression score, we'd get very similar to scores. These are the technologies we use. I won't go into this, but it does take a village of both technological advancements, AI, deep learning, a lot of sort of computational folk we work with around the world to really compress these data sets and to extract meaningful information out of them. And so I'll go to some of the work that's funded through the Cure Cancer Grant. This is really focusing on patient biopsies where we have these on-call biopsies. They're prepared into tissue microarray, single cores per patients or duplicates. And we use a frame of, this is the spatial proteomic data frame, really trying to look at tissue architecture, immune profiling, lymphocyte profiles, immune activation, and we add in sort of functional information and enzymes against metabolic activity. And this allows us to subset deeper into the cell phenotypes and not just at a broad cell class level alone. And so this is the study design where we look at multiple tissue microarrays. We've got over 100 samples in this study. We're able to do the cell typing and generate these sort of flow diagrams, and that metabolic and functional profiling is really important to subset these cell types, especially in the microenvironment. And so this is just a clinical cohort overview. I won't go into this, but it sort of breaks it down by the clinical pathological findings, best objective response rate, and CV6 is clinical benefit at 6 months. These are patients on first-line IO therapy in lung cancer. And so we can look at global compositional differences in the tumor and the non-tumor, but where it gets really interesting is when we look at the functional annotation. So really using single cell proteomics and leveraging the functional and metabolic information. And so we can see the green is high, blue is low. So green is enriched for in the responders, blue is enriched for in the non-responders. And we can see that there are these B cell groups that are high in glucose G6PD activity, so highly metabolically active B cell populations that are found in patients that do well on checkpoint inhibitor therapy. So you can look at what these stars are aligned here. And so then we can break this down by a K2 or a K3. So this is breaking it down by tumor stroma in the K2 or the tumor stroma interface by K3. It allows us 3 dimensions to look at these markers in. And so then we can go into the tumor, the interface, the real sort of action zones or the battlefields, and then also the stroma. And we can again see markers and cell types associated with different functional states. But we can really see that the B cell population is really enriched for in this interface region within the microenvironment. And so then Aaron in the labs really developed this metabolic neighborhood profiling of it, so where we can overlay functional activity and metabolic activity over the phenotype data. So we can look at high metabolic active zones, low metabolic activity by protein, and these intermediate activity zones. And we can plot that directly back onto the tissue over the cell typing data. And so we can look at this across the cohort. This is in different clinical endpoints, such as stable disease, partial response, progressive disease, and complete response. This is at CB6 against RECIST 1.1. And so we can start to look at the metabolic neighborhoods against different clinical endpoints, such as clinical benefit at 6 months, best objective response rate, and so on. And you can really see this activity is really in this mid zone here, where it's sort of glycolysis, ATP synthesis in different tumor interface neighborhoods. And so one of the challenges with spatial data is how do you use the multiple parameters to define predictive biomarkers? And so we use multiple functions, G-cross, Shannon diversity index, you use a neighborhood analysis and sort of measurements of entropy. And so what we've developed on the back end of a Nature Biotechnology paper was the stable feature selection, where we can impute all the spatial parameters into this one feature family, and we can rank this by FDR. And so this is the paper which allows us to do that. So we can look at it in the K2, K3 split and across different metabolic and cell phenotype levels of annotation. And so this is what the hazard ratios look like for OS and PFS. If we were to zoom in on this, this is taking all your spatial interactions that you can measure and then putting it through this model and then pulling out, based on FDR ranking, those that are significantly differentially expressed between responders and non-responders using G-cross, meta-G-cross, Shannon diversity index, and cell proportions. And so we can see this macrophage activity in proximity to areas of granzyme B activity with high metabolic activity is associated with a worse outcome or non-responsive phenotype. But you can see how these other sort of measurements also play a role. Interpreting this is the challenge, and so it's an ongoing challenge in the lab. And so we do this at the whole slide level, not just tissue arrays. This is looking at about 60 markers, 3 to 5 million cells. Every cell within this tissue is profiled. And so we can now contrast tumor stroma interface, metabolic neighborhoods, and tumor density profiles, and we can start to see clear differences with the flavor of functional and metabolic overlay over the phenotype information alone. I won't go into the individual findings here. And so we can overlay the cellular neighborhoods with the metabolic information and tease out these sort of predictors of resistance. We're seeing this activity increase in patients that are resistant to checkpoint therapy, specifically in the tumor cells, but in patients that are responsive or sensitive to checkpoint inhibitor therapies, we're seeing the activity is really in these immune cell types. And so we can look at this over time. This is in responders and non-responders, same site biopsies. And so we can start to classify at a cell level, what are the differences in proximity between a central cell to its neighborhood, between your baseline and your follow-up sample. This is sort of exploratory work that's really early days, but it's giving us insights into what are those predictive signatures here. And also, this is ongoing work in the lab where we're looking at not just protein expression levels, but when you actually have the PD-1 and PD-L1 receptor ligand in close proximity, we can now visualize that signal, right? So this is at 40 nanometers within each distance. And so we can see this proximity ligation assay for PD-1, PD-L1 between the complete responder on the top here, the progressive disease in the bottom here. You can see the zones of activity are different In the partial response and stable disease, this is a diffuse-like signal. We've done this in about 50 patients. We've expanded this, but it's really mapping those interactions and not just relying on the protein distribution across the tissue. And I'll just end with some anecdotes here. I've got a couple of minutes left. This is a whole transcriptome, single cell resolved spatial study. It's just over 600 patients now, all IO, first-line IO. This breakthrough is really important. We're detecting between 500 to 3,500 genes per cell, but spatially resolved signal. We are at equivalency with single cell sequencing. And so this study is ongoing in the lab. We've got multiple people working on this data set, of which Chinwe and Dharmesh are sort of the key drivers of this. This is true whole transcriptome, single cell. It's sort of the evolution of spatial into single cell. We're really excited by this development. And because we have equivalency at spatial resolution, I think the utility of the sort of traditional approaches will decline over time, which is, you're going to see this move into beyond just UMAPs in space. And so this is now plotting that data. This is just a snapshot of that tissue array. We've got 600 patients across this study. This is just one of those TMAs. And I'll end with two sort of exciting stories in the lab. This data came through last week, which was really exciting. 100 patients where we've developed, these are primary early stage lung cancers, resected lung cancers, and we were able to map the individual cell types and then train a model on the neighborhoods within these using this graph contrastive learning model. And so as an input, we know and we have the clinical data of whether the patients had relapse or no relapse as a binary input. And then Aaron came back and he showed us the plots of the cell typing, which is on the top here. This is primary lung cancer, total resected tissue. And then you had a relapse score. And so the relapse score is areas within the primary tissue that have a higher relevance for relapse. And so we sort of brought all the oncologists, the pathologists, the computational folk into the office, and we were sort of hashing this out. Why is it and how is it that we're seeing relapse plotted directly onto primary tissue? And so the idea was, is this logic circular? And everything we've done to date is showing that it's not. We think this is probably learning sort of features associated with tumor aggressiveness. When we've started to overlay markers such as B cells and so on, we can see that it maps to these areas of low recurrence, which is known biology, which is reassuring. We're now overlaying the metabolic information and so on. So using these newer approaches and single cell based transcriptomic and proteomic approaches, I think, these types of sort of discoveries will become really, really exciting. And I will just end with, please don't throw away your tissue blocks. We've been able to look at RNA and protein of 105-year-old tissues. This study was just accepted. The cancer part of this was accepted in Nature Precision Oncology today. This is the differentially expressed proteins between the areas of exudate and pneumonia. You can see the histology separates out on PCA. It does the same on RNA. The RNA is a bit wobbly, but you can see the histology separating out on PCA. This is the protein data of one of those tissues stained for about 21 markers. It made the cover of the Australian magazine, which we're super excited by. This is Lauren, who spent a year in Austria and Germany collecting these tissues. And with that, I'd like to thank the patients that have donated samples to these studies, the incredible team that I get to work with every day, the mentors like Fernando, and numerous Cure Cancer alumni, Orazio, and a lot more. And I welcome any questions. Thank you.